Hello, welcome to my channel. Today I'm going to talk about receptors and cell signaling pathways and the general topics about um, deregulations and how cancer um, is formed when any of these pathways are deregulated. First we're going to start with the quote, which is a motivational quote. Remember to imagine. Imagination will take you everywhere. Next slide. So we have the ligand-gated ion channel receptors. Uh, so uh, don't worry about the names and whatnot, but this is how I organized everything to remember that we have 4 different types of receptors. And I'm gonna briefly introduce these receptors, and I'm sure that you know these receptors because uh, my videos will mostly focus on advanced terms. And if you are not a biology student or if you're in the school level, these videos are not for you. So make sure to know the basic terms of cell biology and molecular biology. And yeah, so we're going to start with the receptors. And we have the acetylcholine receptors. These receptors are ligand-gated ion channel receptors, which means that we need ligand in order to um, have a function, see a cellular response. What is a ligand? Ligand is a molecule which is produced by a cell, and then it affects a different cell, but ligand is recognized by these receptors. And here, a neurotransmitter like acetylcholine can be a ligand, and it can directly bind to the channel. And then ions can come in and cause electrical signaling. And this will change the polarization, and we will see the cellular effect. And I assume that you know that the neurons we have action potential. In the future, I'm going to talk about the neuroscience and details of it, but um, just know that what's happening in neurons, because it's important to know that when axial potential arrives at the axon terminal, we have the calcium entering axon terminal, and the entry of them causes the neurotransmitter to be released. And it binds to synaptic cleft, and then ions enter the postsynaptic cell. So the whole point is that the ligand is bound, and then the ions, such as sodium, potassium, they pass, and then we have the effect. What is important to remember is the structure of the acetylcholine receptor, the GABA receptor, glutamate receptor, and the rest of them. So make sure that you know the structures, that how many membrane spanning components they have. Acetylcholine receptor has 4 membrane spanning component, and the acetylcholine site is on the alpha subunit. That's where the ligand binds to this receptor. And these are the membrane, cell membrane receptors, which means that their location is on the cell membrane. And this is important to know that we have the lock and key model. When ligand binds to this receptor, it will have a perfect binding shape in which the shape of the receptor is exactly compatible with the ligand. And then it's important to know what's allosteric site binding, binding anywhere near the receptor, not on the active side. So it's important to know that when the ligand is not bound, the channels are closed. And when ligand binds, the channels are open and there is the passing. So It's also important to know the size. Acetylcholine receptor length is about 16 nanometers, and it's cationic, which means that ions such as sodium and calcium can go through. So make sure that you know how the neurons work and actual potential. I'm not going to talk about the details of action potential in this video, but in future I'm going to talk about it. But I assume that you have some information about depolarization, action potential, repolarization, the hyperpolarization, and the resting state. The GABA receptors are important for hyperpolarization when there is more negativity and there is it's right before resting state. And it's important to know the structure. They are pentameric receptors, and they need 2 ligands to get activated. And they can also get activated by more ligands, and they have extracellular binding sites, and they also the GABA receptors are created by astrocytes. Astrocytes are actually astrocytes create glutamine, and glutamine creates the GABA receptors. And glutamate receptors have more complex structure. They are tetrameric, which can bind to 4 ligands. It's important to know these structures. Make sure to know that which ions pass through each receptor. For glutamate, we can have sodium, potassium, and calcium passages of these ions, and for GABA receptors, we had the passage of chloride. So it's important to know that CAMP and CGMP receptors have they have 6 TMR. Just like the others, they have the N-terminal, C-terminal binding site is also located in intracellular domain, and the ligand is inside the cell. Remember that the structure is tetrameric, and they can be found in different locations such as retina, hippocampus, kidney, heart, intestine, and so on. And we have the ATP receptors, which are homomeric, and they have 3 subunits, and there are 7 different isoforms of them, and the N-terminal, C-terminal domain, and they are intracellular, and the binding site for the ATP receptors is extracellular. So these were located in the membrane. What about the G protein coupled receptors? G protein coupled receptors are also located on the membrane. They are specific they are actually, from the name, it's obvious that they have the coupling with G protein. And they have 3 subunits, alpha, beta, and gamma. And the binding site for GDP is on alpha subunit. So remember that they need a specific G protein. What's the difference between ligand-gated ion channel receptors and G protein coupled receptors? So the ligand-gated ion channel receptors are fast, really fast, but G protein coupled receptors are not as fast as the ligand-gated ion channel receptors. So that's how when you hurt your hand, the neurons have to sense it and transport the signal in milliseconds. really quick with the ligand-gated ion channel receptors, but G protein coupled receptors take longer. They act in seconds, not milliseconds. It's also important to remember their structure, that they are transmembrane receptors, and they go through the membrane 7 times. The N-terminal is an extracellular area, and the C-terminal is an intracellular area. If you don't know the CEMP system, or phospholipase C pathway, or the way G protein coupled receptors bind to the ion channels, please go and review these, because I'm not going to mention the details. I assume that you already know, and in future we're going to talk about more advanced terms. I'm going to talk about the next group of receptors, which are the enzyme-linked receptors. So one of the main ones, tyrosine kinase receptors, these are very famous, and especially the structure. Remember that they are also in membrane, and the intracellular area is highly conserved. And the ATP binding site is also an intracellular area. And remember that they are inactive when they are in monomeric state, but they become active when they have the dimeric structure. So this is the main feature, that a dimeric structure is the active form of the tyrosine kinase receptor. So how do they do that? They have the autophosphorylation, the receptor phosphorylates itself, and transphosphorylation, when the two subunits phosphorylate each other, the dimers, and then we have the cross-phosphorylation, the receptor. The phosphorylation gets passed on to the other tyrosine kinase receptors. So we have the autophosphorylation, transphosphorylation, and cross-phosphorylation. It's very important to know that main biological function of tyrosine kinase receptors is to respond to growth factors such as EGF, FGF, VGF, NGF, and so on. EGF is important for cell culture, and NGF is to grow nerve fibers in vitro, and so on. They have lots of functions. And so the idea is that when we have the binding of the ligand, we have the conformational change in cytoplasmic tail, and which is the catalytic region, 90% of homology. The homology is not good for us who want to design drugs, because drugs we design won't be as specific as we want them to be. This is important to remember if you study pharmacology. You already know so much about these. And we have the insulin receptor. Insulin receptors are about 51 amino acids, and the active form is T-shape. You probably know that inactive form is U-shape, and you should know how the insulin receptor and insulin pathway works. This is important for diseases such as diabetes, and yeah. But this video has only educational purposes, so don't use these information for any medical or personal usage. So make sure that remember that this is just for educational usage for bachelor students or master students who want to know more about cancer in general, Why are these receptors important for cancer? Why did I start with the receptors? Because um, basically everything is about the cell signaling and these receptors, because any mistake, any tiny mistake in a function of these receptors or the pathways will cause cancer. And it's really important for us to identify these pathways, to inhibit them or stop them. And we're going to talk about the new technologies, about um, new technologies to cure cancer um, in the next videos. So make sure to subscribe and um, follow the next videos. I'm going to talk about the um, next types of receptors and the MAP kinase cascade is also very important, but I assume that you already know it. Just to review, you should know that there is RAS activation, which will lead to RAF, MEK, ERK activation, the phosphorylation, and there will be the activation of transcription factors, and we will have cell proliferation. So you can imagine that if we have more cell proliferation, that is cancer. So mutated RAS is means that it's always active. This will lead to cancer. And also the other cytokine receptors, which are important receptors, and they can activate the transcription. Please study them with more detail, and later we're gonna talk about these in more detail. Intercellular receptors include the corticosteroids and sexasteroids. So the ligands of these receptors have a very specific feature. These ligands are lipophilic molecules. You can imagine why, because the intercellular receptors have a different location. Their location is in cytoplasm level. So you can imagine that this has to be different, the function, the location. When we have the receptor and a steroid complex, they enter the nucleus and there is the binding to DNA, and which will lead to gene transcription and eventually protein synthesis. The main feature of these receptors is the fact that they are in the cytoplasm level. So keep that in mind. And about their structure, I would say that they are about 100 to 500 amino acids, and they also have all the C-terminal region, N-terminal region, and different areas, which I'm sure you have studied. Okay, so what happens when cells receive signals? The ligand is there, the receptor is there, and we have the cellular response, change in phenotype, or any biological function or effect, but actually it's more complex than that. It really is. So we have the cell signaling pathways, which are more complicated than you could ever imagine. Not trying to scare you, but these are the main pathways which you should know about in detail, such as the apoptosis signaling pathway or mTOR pathway, Notch or Wnt pathway, hedgehog pathway. Like, you should know where they are found and where they function and what do they do. You should know that Wnt beta-catenin signaling pathway is important in stem cells. We're gonna have so many videos about stem cells in future, and you should know that hedgehog pathway is for the embryonic development. And of course, it's also very important to know the DNA damage response, because when we have damage in our DNA, there is a response to that. It gets fixed. If it doesn't get fixed, then this will lead to cancer. So all of these pathways are really important. Try to know them off by heart, although it might sound a bit difficult. But okay, so what? Like the whole thing that I talked about. We have the cell, this gray one, which you can see here. This is the cell which has the ligands, the purple ones. So we have the cell here. And also, I'm going to introduce the paracrine signaling to you. The paracrine signaling happens when two cells are near each other. As you can see, the autocrine signaling is when the same ligand which is produced by the cell affects the same cell. So if it activates itself or it binds to itself, then it's autocrine. And then we have the response. So I'm gonna talk about the fabulous central dogma we have here. So what? This whole signaling thing, like a general, very general, brief introduction. So when the ligand binds to the receptor, here you can see the ligand, and it's completely clear that we have some connection here. You can see it. There are lots of molecules and proteins who help with forming this complex. And then we will have the activation, and this activation will lead to gene transcription. And we will have mRNA, the messenger RNA, which will have to exit the nucleus. Then we have RNA splicing before exiting the nucleus. RNA splicing means that the mRNA has to become mature so that when it leaves the nucleus, it doesn't get degraded. So mRNA becomes mature, RNA splicing, it leaves the nucleus, and it gets translated into protein by ribosomes. And this is the fabulous central dogma. And this protein, which is created, can have so many functions. It can turn into a new receptor or anything, literally any function. And we also have the endocrine signaling, which you can see that the ligand, which is produced, or the hormone, is transported through blood. It's going there, and then it will eventually reach the cell, and it's going to leave its effect. So it's important to know we have three different forms of cell signaling: paracrine, autocrine, and endocrine. Okay, so I would say that's it for today, and thank you for watching. Next video will be a brief introduction about cell cycle, DNA damage response, and how important it is in cancer. Make sure to subscribe, study hard. See you soon. Bye.